So you want an explanation of glycolysis? Delightful. I'm a writer, but even I can appreciate the simple poetry of glucose being slowly dismantled for energy. Have you ever wondered how a single sugar molecule can kickstart the cellular equivalent of a tiny power plant? Glycolysis is basically a 10-step biochemical relay race in the cytoplasm that converts one glucose, a 6-carbon sugar, into 2 molecules of pyruvate, each with 3 carbons. Along the way, the cell nets a small but crucial payoff: 2 molecules of ATP, the cell's currency, and 2 reduced NADH molecules. Think of NADH as IOUs full of potential energy. Why 10 steps and not one grand dramatic transformation? Because subtlety matters. Each enzymatic step lets the cell regulate flux, harvest energy efficiently, and funnel intermediates into other pathways. Also, enzymes like hexokinase and phosphofructokinase act as gatekeepers. Phosphofructokinase in particular asks the philosophical question, should we feast or conserve? It's highly regulated by the cell's energy state. Let's break it briefly into phases. First, the investment phase. The cell uses 2 ATP to phosphorylate and reshape glucose into a form that can be split. Sounds wasteful. Why spend energy to make energy? Ever invest in a nicer suit to get a better job? Same principle. Then comes the payoff phase. The 6-carbon intermediate splits into 2 3-carbon molecules that are processed to produce 4 ATP and 2 NADH, leaving us with pyruvate. What happens next depends on oxygen availability. Under aerobic conditions, pyruvate can enter mitochondria and be fully oxidized for far more ATP. Under anaerobic conditions, it's reduced to lactate or ethanol to regenerate NAD+, letting glycolysis continue. Isn't that elegantly pragmatic? So what intrigues you more, the regulatory finesse, allosteric control, feedback loops, or the metabolic fate of pyruvate? I have refined tastes unlike most, but both are delicious topics.